Let's take a quick minute and go over the concept of tonicity. So when we talk about tonicity, we are talking generally about the concentration of solutes outside the cell compared to the solutes inside the cell. So let's use an example here where I have 3 beakers and 3 cells. Now let's assume that all of the cells have the same solute concentration. So inside my first cell, I have a 9% solute concentration. And let's assume that the rest of my cells are the same. So they also have 9% solute. Now if you think about a cell that's filled with fluid and that also has these solutes kind of floating around in that fluid, it's a little bit like a chocolate chip milkshake, where the milkshake itself makes up the fluid in the cell, and the chocolate chip chunks are the solutes. If I have more chunks of chocolate chips, then there's less room for milkshake. So if I have more solute in a cell, there's less room for water. Let's assume that this first cell is going to be put down into a solution that is just pure water, 0% solute. The second cell is going to be put into a solution that is 9% solute, or the same as the inside of the cell. And my third cell is going to be put into a solution that's 24% solute. So it has more solute than inside the cell. So once I move my cell into that solution, now, in the first case, I have a solution that's 9% inside and 0 outside. So there's more water outside the cell because there's less solute taking up space. Water, like any other molecule, always wants to move down its concentration gradient. So the water will want to go to where there is less water, or it will go into that cell. In the next case, I still have 9% solute inside and 9% outside. So there's the same amount of water on both sides also. So the water will go into the cell, but it will also go out of the cell, so there will be no net movement of water. In the third case, I have 9% inside and I have a much higher concentration of solute outside. Well, if there's more solute out here, that means there's less room for water. So the inside of my cell has more water. The water will want to go to where there's less water. So with the first cell, I put that cell into a solution that was lower than the inside the cell, so that solution was hypotonic to the cell. In the second case, I put my cell into a solution that was equal, so that solution is isotonic to the cell. And in the third case, I put the cell into a solution that had more solute, so that outside solution was hypertonic to that cell. So in a hypotonic solution, water will move into the cell and the cell will expand, or it could lyse or burst. In the second case, in an isotonic solution, everybody's happy. So this is what animal cells like, or cells that don't have cell walls. We like an isotonic solution. In the third case, my cell was put into a hypertonic solution, which drew the water out, so my cell shriveled up, or it crenated. So think of this like putting a bunch of salt or sugar or spice on a steak and leaving it overnight. It's going to dehydrate that steak or dehydrate that food that you put it on. That's how you actually make beef jerky. Our cells like isotonic solutions. Plant cells like hypotonic solutions. So when you water your plants, you're just giving them plain pure water. Water goes into the cells. But the reason that they don't lyse is that plant cells have a cell wall. and that rigid cellulose cell wall prevents it from bursting. So use this concept here to help you answer those short answer questions about tonicity.